Scientists have uncovered a new blood type, and right now there's only one person in the world that has it.
An urgent call to step up and save a life. A Sarasota blood bank is running low on O-negative blood.
For more than a century, scientists and medical experts have been fascinated by the mysteries of human blood types, particularly the elusive characteristics of type O blood. Among the most crucial and universally recognized blood types, type O has stood apart, not just because of its compatibility across all other types in transfusions, but also due to its evolutionary, genetic, and immunological quirks that have puzzled generations. Now, after decades of research, a groundbreaking scientific breakthrough has finally unraveled the enigma surrounding type O blood, stunning the global medical and scientific communities. This revelation not only clarifies the origin and function of type O blood, but also opens entirely new avenues in medicine, genetics, anthropology, and even biotechnology. Before we start, smash the like and subscribe buttons for more updates. The story begins with a focus on the ABO blood group system, one of the most important classifications in transfusion medicine. The system, discovered in the early 1900s by Karl Landsteiner, defined blood types A, B, AB, and O based on the presence or absence of antigens on red blood cells. Type O stood out because it lacked both A and B antigens, making it the universal donor in emergencies. However, despite its practical use, type O's lack of antigens also presented a biological mystery. Why would nature produce a blood type that, on a molecular level, appears to be lacking something that all others possess? Over the years, scientists speculated about this absence. Was it an evolutionary adaptation to disease, a fluke of human genetics, or perhaps something more complex hidden in the code of our DNA? Early genetic studies showed that type O blood was caused by a specific mutation in the ABO gene, which effectively turned off the production of the A and B antigens. This mutation was passed down over generations, spreading across populations globally. Yet, even this discovery didn't satisfy the deeper questions. Why did this mutation occur, and what benefit, if any, did it provide? The answer finally emerged from a collaborative international study involving cutting-edge genomic analysis, synthetic biology, and evolutionary modeling. Teams from institutions across North America, Europe, and East Asia combined efforts to conduct an unprecedented analysis of the ABO gene across multiple populations, including ancient human DNA samples dating back tens of thousands of years. What they discovered was nothing short of astonishing. Type O blood was not merely a mutation, it was a deliberate evolutionary safeguard, a biological strategy developed in response to one of humanity's most ancient and lethal enemies, pathogenic microorganisms. Their findings revealed that the absence of A and B antigens in type O individuals significantly reduces the susceptibility to certain kinds of bacterial and viral infections, particularly those that use blood group antigens as docking mechanisms to invade cells. These pathogens, such as Helicobacter pylori and certain strains of Plasmodium which cause malaria, have evolved to exploit the presence of A or B antigens. Individuals with type A or B blood are more likely to be targeted due to the biochemical architecture of their red blood cells. Type O individuals, lacking these entry points, often experience less severe symptoms or even avoid infection altogether. More impressively, the research traced the mutation's origin back to a critical period in human prehistory, roughly 100,000 years ago, during a time when early humans were undergoing severe population bottlenecks due to climate shifts and widespread disease. The type O mutation seems to have offered a survival advantage during pandemics in ancient times, allowing carriers to survive outbreaks that decimated those with other blood types. As a result, the mutation spread rapidly across continents, becoming particularly dominant in regions where such diseases were historically endemic. Yet, the revelations did not stop there. Scientists also discovered a previously unknown layer of immune regulation tied specifically to type O blood. Using state-of-the-art molecular imaging and AI-assisted protein modeling, researchers identified that type O individuals possess a slightly altered configuration of H antigen, a precursor molecule that would otherwise become A or B antigens. This unique structure not only limits pathogen entry, but also subtly enhances the body's innate immune response by allowing immune cells to detect certain threats earlier. The result is a faster mobilization of white blood cells and a more efficient inflammatory response, something that has long been observed anecdotally in clinical settings but never fully explained, until now. This new understanding transforms the entire approach to immunogenetics. It's no longer just about what antigens are present on red blood cells, but about how the absence of these structures reshapes the body's defense systems from the ground up. The absence of A and B doesn't mean a deficiency. It's a redesign, a recalibration of immunity itself. Scientists are now exploring how this discovery can be used to engineer artificial blood for transfusions, tailored organ matching, and perhaps even vaccines that mimic the immune profile of type O individuals to confer broader protection. What adds another layer of fascination to this discovery is how it redefines our understanding of human evolution and migration. The genetic evidence suggests that the O-type mutation arose independently in multiple populations, a phenomenon known as convergent evolution. This means that natural selection repeatedly favored the same mutation in different human groups, underlining its extraordinary survival advantage. The recurrence of this adaptation across geographic and genetic boundaries reinforces the idea that type O blood was not just a genetic fluke, but a product of evolutionary necessity. In parallel, anthropologists are revisiting ancient population models, reassessing how the prevalence of type O blood may have influenced social structures, survival during famines, and resistance during ancient plagues. Archaeogenomic reconstructions show that groups with higher proportions of type O blood were often the ones that repopulated after major die-offs, creating a cascading genetic legacy that still echoes in modern gene pools. These revelations are poised to reshape the historical narrative of human survival and dispersal across the planet. The implications of this discovery also stretch far beyond the confines of academia. Biotech firms and pharmaceutical giants are already looking into applications of these findings. One major pharmaceutical company is exploring synthetic analogs of the unique H antigen configuration found in type O blood as a template for antiviral compounds. Another research team is developing a CRISPR-based therapy to replicate the immune-boosting effects of type O blood in individuals with other blood types. The dream of engineering more robust immune systems, once relegated to science fiction, now sits squarely within the realm of possibility. Furthermore, the revelation has stirred renewed interest in personalized medicine. Doctors may soon tailor treatments based not just on blood type compatibility, but on deeper genetic profiles rooted in how the immune system has evolved to respond to environmental pressures. In time, this could revolutionize everything from organ transplantation protocols to cancer immunotherapies, where individual immune profiles are critical to treatment success. Public health experts, too, are examining how this knowledge might help predict disease spread in future pandemics. By mapping populations with high concentrations of type O blood, they may be able to model relative risk more accurately and deploy resources more effectively. Vaccine developers are also exploring how antigens interact with different blood types to refine their formulas for broader, more equitable efficacy. On a broader scientific scale, this breakthrough exemplifies the power of interdisciplinary research. It took not just geneticists, but also immunologists, evolutionary biologists, data scientists, and epidemiologists working in concert to decode the mystery of type O blood. This synergy of fields is likely to become the blueprint for tackling other complex biological puzzles, as no single discipline could have unearthed the full scope of this discovery on its own. There's also a psychological and cultural dimension to this moment. For centuries, blood types have carried symbolic and sometimes mystical connotations, especially in cultures that assign personality traits or compatibility based on blood groupings. While those beliefs are not grounded in science, the new findings may offer a more nuanced understanding of how blood types genuinely affect human physiology and health outcomes. The realization that something so foundational to our identity holds deeper biological truths offers a profound reminder of how much we still have to learn about the human body. One of the more curious findings from this research concerns pregnancy and reproductive biology. Researchers have long known that women with type O blood sometimes face higher rates of certain pregnancy complications when carrying fetuses with other blood types due to immune reactions. But the new insights into the H antigen structure have revealed that the immune system's response is not merely a rejection mechanism, but a finely tuned regulatory process that influences fetal development, placental function, and even maternal fetal gene expression. This has sparked a fresh wave of studies into prenatal health and how this subtle form of immunological dialogue shapes outcomes from conception onward. In the field of transfusion medicine, the consequences are even more immediate. With the ability to synthetically mimic or convert blood types using enzymes and gene editing, hospitals may one day maintain universal type O blood supplies that are lab-grown, pathogen-resistant, and immunologically optimized. Such an innovation would mark a quantum leap forward for emergency medicine, disaster response, and military field care. It would eliminate shortages, reduce dependence on donors, and potentially save millions of lives annually. This also reframes long-standing medical debates about blood type-related disease susceptibility, such as the link between type O blood and lower clotting factors or increased risk of ulcers. Instead of viewing these traits as isolated quirks, scientists now see them as components of a broader immunological strategy encoded deep in our genetic history. These features are no longer considered liabilities, but trade-offs in a complex biological equation optimized for survival under specific environmental conditions. Even fields like forensic science are benefiting. The unique protein signatures linked to type O's antigen profile can now be used to improve accuracy in DNA matching and crime scene analysis. It enhances the granularity with which investigators can identify not only an individual's identity, but their potential health markers and genetic predispositions, further refining the scope of forensic evidence in judicial proceedings. Beyond medicine and science, this breakthrough taps into something profoundly human: our desire to understand who we are, where we come from, and why our bodies function the way they do. The mystery of type O blood, long dismissed as merely a convenient donor status, now stands revealed as a linchpin in the story of human survival. It reminds us that evolution doesn't always add complexity. Sometimes it subtracts, streamlining the blueprint of life for resilience, not just variety. Every so often, a scientific discovery comes along that does more than advance knowledge. It reframes assumptions, challenges paradigms, and opens new doors not only for research but for imagination. The solution to the mystery of type O blood does just that. It bridges ancient genetics with modern technology, primal survival with futuristic innovation, and individual biology with collective destiny. It's a moment that signals not just a scientific victory, but a deeper awakening to the extraordinary intelligence of nature's design. A design written in the invisible ink of our own blood. The implications of this discovery ripple outward into sectors of science and society that, until now, seemed only tangentially related to hematology. One of the most unexpected fields to benefit is regenerative medicine. As research continues into stem cell therapies and tissue engineering, understanding the micro-level mechanics of blood compatibility becomes vital. The clarity now gained about type O blood, especially its molecular profile and evolutionary purpose, enables researchers to tailor cellular therapies with far greater precision. When growing tissues or organs in the lab, scientists must consider immune rejection. A major hurdle has always been ensuring compatibility with a patient's immune system, particularly regarding surface markers like blood antigens. With the full characterization of type O's unique traits now available, labs can begin constructing neutral tissue, cells engineered to emulate the antigen-absent profile of type O blood. These neutral tissues, being less likely to provoke immune responses, stand to revolutionize transplant medicine. Not only would organs be more compatible across broader populations, but the risk of rejection could be dramatically reduced without aggressive immunosuppressive drugs. In cancer research, the revelation adds yet another dimension to how tumors interact with the immune system. Scientists are now re-examining how antigen presence or absence affects tumor growth and immune evasion. There is early indication that the distinct cellular environment in type O individuals may play a role in either suppressing or accelerating certain cancer types, depending on how cancer cells use antigens to mask themselves from the immune system. This knowledge may pave the way for blood type-specific cancer therapies, introducing a previously untapped biomarker for personalized oncology treatments. microbiome research is also feeling the tremors of this breakthrough. The bacteria and microorganisms that inhabit our digestive tract often interact with host cells based on available surface markers, including blood group antigens. With the newfound understanding of how type O's absence of antigens influences immune interactions, researchers are revisiting how gut flora behaves in these individuals. Preliminary studies suggest that type O individuals may harbor a microbiome composition distinct from other blood types, potentially influencing digestion, metabolism, and even neurological conditions via the gut-brain axis. Even the field of pharmacogenomics is undergoing a shift. For years, doctors have tailored prescriptions based on genetic markers related to liver enzymes and metabolic pathways. But with these new insights, blood type, especially type O, might join the roster of relevant genetic indicators. Type O individuals could exhibit different metabolic responses to certain medications, particularly those processed through immune-modulated pathways or those that interact with blood-borne proteins. Understanding this can help doctors calibrate doses more accurately and avoid adverse drug reactions. Another frontier influenced by the type O breakthrough is chronomedicine, the study of biological rhythms in relation to disease and treatment. Early evidence indicates that individuals with different blood types exhibit subtle variations in circadian gene expression, impacting how the body repairs itself or responds to pathogens over a 24-hour cycle. Type O, in particular, appears to maintain a slightly elevated baseline of certain inflammatory markers, which may correspond to a more vigilant immune rhythm. This opens up opportunities for optimizing the timing of immunotherapy, vaccination, and drug delivery based on an individual's blood type to maximize efficacy. Nutritional science is also absorbing the impact. For years, the blood type diet had existed on the fringes of mainstream science, dismissed as pseudoscience due to lack of mechanistic support. However, with solid molecular data now confirming unique immune and microbiome traits associated with type O blood, researchers are cautiously re-evaluating whether tailored dietary recommendations based on blood type might actually have merit. While it's premature to endorse fad diets, a targeted nutritional plan that supports type O's distinct microbiome and immune environment may emerge from legitimate scientific inquiry. This reawakening of interest in blood type as a key to understanding the body has also rejuvenated efforts in neuroimmunology. There's mounting curiosity about whether blood type, particularly type O, has any influence on the immune-brain relationship. Since type O individuals seem to possess a slightly more reactive immune system, researchers are investigating whether this translates to differences in neuroinflammation, cognitive resilience, or even susceptibility to neurodegenerative diseases like Alzheimer's. Although still in its early stages, the connection between immune response profiles and brain health. For more than a century, scientists and medical experts have been fascinated by the mysteries of human blood types, particularly the elusive characteristics of type O blood. Among the most crucial and universally recognized blood types, type O has stood apart, not just because of its compatibility across all other types in transfusions, but also due to its evolutionary, genetic, and immunological quirks that have puzzled generations. Now, after decades of research, a groundbreaking scientific breakthrough has finally unraveled the enigma surrounding type O blood, stunning the global medical and scientific communities. This revelation not only clarifies the origin and function of type O blood, but also opens entirely new avenues in medicine, genetics, anthropology, and even biotechnology. Before we start, smash the like and subscribe buttons for more updates. The story begins with a focus on the ABO blood group system, one of the most important classifications in transfusion medicine. The system, discovered in the early 1900s by Karl Landsteiner, defined blood types A, B, AB, and O based on the presence or absence of antigens on red blood cells. Type O stood out because it lacked both A and B antigens, making it the universal donor in emergencies. However, despite its practical use, type O's lack of antigens also presented a biological mystery. Why would nature produce a blood type that, on a molecular level, appears to be lacking something that all others possess? Over the years, scientists speculated about this absence. Was it an evolutionary adaptation to disease, a fluke of human genetics, or perhaps something more complex hidden in the code of our DNA? Early genetic studies showed that type O blood was caused by a specific mutation in the ABO gene, which effectively turned off the production of the A and B antigens. This mutation was passed down over generations, spreading across populations globally. Yet, even this discovery didn't satisfy the deeper questions. Why did this mutation occur, and what benefit, if any, did it provide? The answer finally emerged from a collaborative international study involving cutting-edge genomic analysis, synthetic biology, and evolutionary modeling. Teams from institutions across North America, Europe, and East Asia combined efforts to conduct an unprecedented analysis of the ABO gene across multiple populations, including ancient human DNA samples dating back tens of thousands of years. What they discovered was nothing short of astonishing. Type O blood was not merely a mutation. It was a deliberate evolutionary safeguard, a biological strategy developed in response to one of humanity's most ancient and lethal enemies, pathogenic microorganisms. Their findings revealed that the absence of A and B antigens in type O individuals significantly reduces the susceptibility to certain kinds of bacterial and viral infections, particularly those that use blood group antigens as docking mechanisms